this beneficial effects in Parkinson's disease. However, a number of studies have shown, including the European study, and the Millennium study is actually from USA, and they have shown that if the animal models or even humans, is one of the very, ubiquitously present in many of these herbs used, which is present also in chocolate in a small quantity. And this itself is a dopaminergic neurotoxin. So, the claims are not from just like that, but it is with a basis. And then anti-Parkinsonian moieties are also available in these drugs. So let us look at that. So some group has carried out a very systematic study wherein from the Ayurveda hospitals, which is from the DGHS, there is this central government health departments, the doctors prescribe these seeds or herbs, and these are collected through the hospital, and then they have been validated by a pharmacognosist about the herbal identity, and then this has been cleaned and powdered and extracted with solvent system. And then this has been characterized and found, and then what they have done is they have removed the L-DOPA content. Because if other than L-DOPA there is something else which is contained in this preparation, that is giving the beneficial effect. So L-DOPA has been removed from this preparation and then shown that, several of them has been analyzed and analyzed in vivo and in vitro models. And a couple of compounds have shown that by correcting mitochondrial electron transport complex dysfunction, the same molecule can stabilize the mitochondrial fission, fusion, and it can also stabilize mitochondrial respiration that is reduced in Parkinsonism, and by upregulating the levels of cyclic AMP in the brain, as well as upregulating GDNF and tyrosine hydroxylase in the brain, and it also has a quality to oligomerize alpha-synuclein. So, based on these results in the preclinical study, the compound that is isolated from one of the herbs is stated to be a good molecule. Now, what do we want? This is why I'm presenting this as a first case study. We need to validate this in a different laboratory, independent of our influence. This is absolute necessity. Case study 2A. I have got two cases, which is very small. I'm not going into details. One of the things that we are doing is that we just collect samples from the animals, dissect out with laser microdissection microscopes and everything, remove the ventral tegmental area, which is more towards the psychiatry problems, nothing to do with the movement. We take substantia nigra pars, compactor region, which is actually involved in movement disorders, and do the proteomics. So we do that in the MBTP model too. Then what? We need to validate that in Parkinsonian brain, postmortem samples. There's nothing else can be done. And we have a brain bank in India which is in NIMHANS, but it is not enough because much of the tissue has been utilized. We do not get VTA. Sometimes we don't get SNPC. So then what we have done is, we need to validate this. Yes. Then what we have done is, we have done this is the proteomics study I wanted to show. Especially I wanted to show you that two of the proteins which has been identified in the ventral tegmental area which is high, at the same time the substantia nigra area which is very low. You can see that the proteomics is different. The proteins are different. Just see this almost 2 to 3 fold in ventral tegmental area. And this has been established. I don't want to get into details. And look, another protein, which is HSPA8 as well as PHB, the same, its expression is about 4-fold increased in the ventral tegmental area, not in the substantia nigra. Does it have anything to do with Parkinson's disease? That's case study 2B. We have created a lot of hybrids from the Parkinsonian as well as control patients because we don't have enough of postmortem brain samples. So what we have done is we have utilized the mitochondrial genome in a control population and the mitochondrial genome in a patient population, PD patient population, and created hybrids. And this can be just propagated and it can be used extensively. And this can be made into neurons, differentiated into neurons, and we can do a lot of studies. See this, such an example here. You can see the cells of, this is the control. The blue one is the control hybrid, and the Parkinson's hybrid is this. You can see. So such a dysfunction, which is available in the brain of the Parkinsonian patients, can be reproduced in these cell lines, which have been created by fusion of the mitochondrial genes introduced into a Rho-0 cell, which doesn't have any mitochondria. Now, my question is, we do have created control and Parkinson's disease hybrid from the Indian population. Proteomics of control and PD hybrids are assessed. Drugs are now being screened using these cell lines. Can we collaborate in this area? So, in a nutshell, we have got two more. I just want to say independent validation of claims made in Indian labs. The second one is sharing of postmortem brain tissues for research. And the third one is sharing of transgenic animals for, or tissues, brain or tissues. Of course, then there can be exchange visits, collaborative programs, joint graduate research programs leading to award of degrees from both the institutions, both the institutions from both the countries. Thank you.
Yes.
But could you identify?
Oh, I'm sorry. Apologies. My name is Dr. Kamlesh Asotra, and I'm a professor of biosciences, translational research, and regenerative medicine at APU Satyavok. My suggestion would be, if you're looking at some of these potentially useful Ayurveda or homeopathy-based, therapeutics, could one also do a prospective study in a select number of patients for maybe a year or two, just using the run-of-the-mill functional MRIs? And I'm sure we have plenty of functional MRIs in India. Won't you think?
Yeah. You need to look at the history. For example, earlier we used to use, so from there we have got aspirin, which is about 115 or 120 years ago. Even now we are using aspirin. So we want to contribute. That means, we need to isolate, molecules that are extremely good, very potent, and probably will not be using other molecules which are contained in the herb, which is bad for us. So that should be the modern way. And this is approved. This is already proved. I have given you a typical example of aspirin.
Premier tertiary referral center for several disorders. So he will actually present us with the clinical side and what might be their needs in making our basic research and translational research more relevant to the patient.
Hi, I'm Sumit, for about 9 years. And in 2009, I switched over to the private sector, and now I'm more into clinical evaluation of patients. My primary area of interest is Parkinson's disease and other movement disorders. An answer to your question, sir. We have Professor Schuntek from Germany, and we are in collaboration with him. He is a German individual who has got about more than 250 publications on movement disorders and basic sciences, and now he is into Ayurveda. So he has evolved a full center for Ayurvedic research in Germany, in Munich, and we are in collaboration with him doing clinical research on Parkinson's disease using alternative medicines. Now he has a full evaluation protocol and a treatment protocol in which the patients are taken in the hospital and they are subjected to various aspects of interventions in terms of Ayurveda. And this is basically a very scientific basis in which we do a UPDRS scoring of the patient, we do a imaging of the patient, we do a quality of life assessment of these patients before and after the intervention on Ayurveda. And he has found some amazing results in terms of the non-motor complications which we see in advanced Parkinson's disease. His opinion is that Parkinson's disease is not a disease of the brain to begin with. His opinion is that it is a disease of the intestines to begin with. He feels that the first symptom of Parkinson's is not actually the rigidity or the tremors, it is the constipation, which probably evolves much, much earlier than the motor symptoms of Parkinson's. And it is even a proven fact that amongst the preclinical symptoms of Parkinson's, it's probably that constipation and anosmia would be the ones which are probably much, much earlier than the clinical segment of the disease itself. So we are into collaboration with him, and we have probably the only center for a dedicated Ayurvedic treatment in the northern part of the country. So that is one part. I am a hardcore clinician, and I have sort of left clinical research for the time being because I am in the phase of stabilization and consolidation my institute. This is not evolving of new research. What I think from the clinician's point of view, what is more important is implementation of the existing research. Because the gaps which we find in India specifically are that one, the patients are not aware about a disease, the advances of the disease. We have all the medicines available, but the point is the education of the patient is extremely important, which is woefully lacking. So a patient education program has to be evolved, which tells these patients that this is disease in which your body is like a motor car, in which if you don't put in petrol every day, it's going to stand. And when your car comes into reserve, a next set of petrol has to be put in. So these people don't know that the medicines have to be taken lifelong. They don't know that these medicines are not a cure for Parkinson's disease. They are in fact just a symptomatic improvement. They do not know that what are the dietary modifications which are required for taking these medicines with the food, away from the food. What are the physical aspects of these medicines? What are the physical treatments which these patients have to take in? Then, obviously, the moment they find some side effect of the medicines, they would stop these medicines. And again, they would come back that I'm not as good as I should have been. The third component is that the available treatment of deep brain stimulation, which is an excellent modality to improve the clinical quality of life of a patient, is not even known to most people. Despite the best of our efforts, we are into, non-government organization type of a situation also, in which we have made a group which is a support group for Parkinson's patients, in which we try to tell these patients that what is the disease, why is it caused, what are the physical aspects of it, how do you treat it, and then we also tell them about deep brain stimulation. But unfortunately, I would say that less than 1% of the total patients of Parkinson's disease actually would get a DBS done on their own, even with significant counseling, whereas more than 30% actually need it at one particular time. And Dr. Rupam is here. They're doing about one case a week. And we should understand that there are only 5 or 6 such centers across the country. So what is binding for this intervention are 2 segments. One is the awareness of the people that this disease is going to show a significant improvement by DBS. That is one thing. The second thing is the cost of the equipment. So where a collaboration might help us is to develop an indigenous protocol or an indigenous equipment for deep brain stimulation, which would be probably produced in India or maybe modified according to Indian requirements, and then therefore thereby bringing down the cost of those things. Then the second point is that research in India is not economically very viable. So that is something which needs to be understood, and this is a very, very important aspect. The type of funding which is required for research is phenomenal. and the type of funding which the government sector provides is abysmal. So there is a huge gap between the returns which you get on research front and the returns which you get on a clinical front in our country. For example, when I was in All India Institute of Medical Sciences, I would hardcore into clinical research apart from clinical activities. The moment I switched over from the government sector to a private sector, the difference which I find is I am too much occupied with patients. And when I want to do some clinical research, the clinical activity tends to take a backseat, which would not be permitted for economic reasons. So I think there should be a compartmentalization in the private as well as the government sector of the Indian subcontinent, where not only there should be a definite incentive-based promotion for clinical research apart from the clinical activities. So that is the second part which needs to be evaluated. And the third thing is that there is a lot of activity going on in terms of stem cell research in our country. However, the actual research is very, very low. So one is that it has to be told to the people that stem cell is not a, panacea for all types of neurological or neurological disorder or non-neurological disorders. So it has to be told to the masses and the classes that stem cells are an investigative tool at best. They have to be told that these stem cell parlors, which are growing up in different countries and different parts of the countries, are not actually treatment options. They are only a way of utilizing the emotional quotient of a patient who is running from pillar to post with a disease which is not curable. So what I think is a third component of collaboration should be a stem cell collaboration in which there is a clinical center which can take part of the clinical part and a non-clinical center which can take care of the basic science part. And then they collaborate in terms of provision of differentiated cell lines in terms of stem cells and an area which can use it for clinical purposes. So that is my submission as a clinician. I'm sorry I'm extremely short in terms of the duration, but that's what I felt. Thank you very much.
Maybe I will start by a pragmatic question. How, in fact, could we help or how could we collaborate for the information of the patients? Because it's also a great need in our country, in fact. We are not, as in the US or Canada, countries in which we are used to speak with the patients. In fact, there is a relatively strong barrier between the patients and the physicians. So it's maybe I think we should try to collaborate on this. And there is one possibility, which is the Alliance for the World Alliance for Parkinson's Disease, and there was the World Parkinson's Disease Congress with more than 1,000 patients attending the meeting in Montreal 2 weeks ago. And I think that these are ways, in fact, to inform the patients. So on that ground, we might collaborate.
No, we are already very, very seriously into it. When I was in the All India Institute of Medical Sciences, we used to organize one event annually in which we used to invite all the patients with the Parkinson's disease on one single day. And there used to be a half-day program in which we used to tell these patients as to what is Parkinson's, why is it caused, the treatment, the surgery, everything. And then these patients used to have an interaction with the experts, the physiotherapy, the speech therapy, everything was discussed in detail. And then we all used to have a lunch. So it was more of a socio-scientific gathering. Now, that tradition I have carried forward in my institute today as well. So when I had started my practice, at that time I didn't have much patience. So what I did was that I approached the senior citizen forums in my city. And in those senior citizen forums, I invited the decision makers in the communities to come and have an interaction with us. Now, in the next year of my practice, we started having patients. So then we invited these patients for an out-based camp. And then we took about 25 families together for a meditation program across the country. So that is the type of work which we are doing. But I think a single day of activity is not going to make any difference. I think it has to be a continuous program, and I call it as a spoke and a wheel system, in which we create a wheel in which there is a center person who can be a guide to guide others who in turn can be becoming the center of the wheels, and this can spread across the country.
Thank you. We will move on, and it's my pleasure now to introduce Professor City with a big red circle in south of France, and Olivier Blin has created a European network which is called Pharmacog. It's a big success, and he spent 2 years and a half in Shanghai running the Center for Neurodegenerative Disorders for GSK, and now he's back to the academic in France. So you will give us the view of the pharmacologist and perhaps also a slight view of the industry.
Thank you, Etienne. It's my pleasure being here. You didn't mention something that is of importance. In 2010, I spent 1 year in India, in Hyderabad, so working with the Indian group, and especially with Rupam here. And Rupam, we also explain how we developed the collaboration between France and India. My topic today will be the clinical research. How is it organized in France, and how we can benefit for the translational medicine. And as you know, there are many challenges in the neuroscience diseases for first understanding the biology. And you recently heard the CEO of one big company saying that since academic research is not updated on the pathophysiology of neurodegenerative diseases, most of the companies will disembark from these boats. And to the end, it's also difficult for us to determine as a clinician what will be the treatment duration, how to treat the patient, and what is the therapeutic window. If we try to give an overview of the disease, not on the symptomatic treatments, but on etiological treatments, we have to determine what we want to target, pathogenesis or pathophysiology. And as Etienne already mentioned, we can target, I don't know if there is... I could show here. We can target or study the genetics and environment. We can focus on the primary damage, but also on the secondary damages, and to have markers for pathophysiology and biology up to symptoms. And then when patient fulfill the classification criteria, we can include him or her in a clinical trial. But we also have to understand that after some progression of a disease, there are different factors, different mechanisms that take place that can modify the treatment and the way we can understand the disease. And for neuroprotection or neurorepair, the therapeutic window opportunity might vary, and we can address here with prevention some genetics or environment, but also we can address later on for treatment either the primary damage or later on the symptoms. And of course, the idea will be completely different. I try to summarize on this slide what might be the direction of travel we can take together. That is melding traditional and novel approaches. And I will focus on translational biology and discovery medicine, because we clearly need to have the integrated thinking to bridge between clinical research and basic research. And as Etienne already mentioned, to be sure of what is the clinical tractability with the biomarkers, and also building some cohorts, both in France and in India, that are fully useful if we get first the informed consent of the patient, and then the clinical phenomenology, and then the biology, the biomarkers, the imaging, and the progression of a disease. And if we are able to do that, then we will be able to identify the right disease and the right patients. But also we have to be able to design the best trials. That is, we have to build, especially for drug development, on 3 or 4 cornerstones, to develop breakthrough techniques, to develop the biomarkers, to optimize the clinical trial design, and also to build integrated platforms, both in France and India, that are able to collaborate, including e-health, and India is very well known for these capacities. And also to don't forget that we have to comply with the regulatory requirements in both countries, and also vigilance, that is safety, that is pharmacovigilance, that is also to guarantee the safety of the patient. Industry and academic have somehow overlapping interests, but that might be a little bit different. When I was in the industry, my goal was to shorten the timelines, to allow to get decision-making tools, to discharge the risk of progression of a treatment, and also to control the cost within the business cycle. When we are in the academic field, usually we focus on expertise, on the benefit-risk ratio of treatment for a patient. We focus on education and training, but also on integrated plans, management and cost. This is quite new in our way of thinking. Sorry, there is a copy here. And finally, also of valorization. Because if we want to get the support for our research, and if we want our research to benefit to the patient, then we have to think about the value, the added value of our work from the beginning of our activity. Key numbers in France, so I don't have circles, I have squares, but it's the same. And the key numbers here, you can see, there are different organizations, a budget, that you can see here, and I will just emphasize one aspect that are the competitiveness clusters, because I've been at the board of the French Minister of Health, representing the minister, to build these clusters, that is organizations that are able to link the academic people, the education, the research, the clinic, with the SMEs and also the large group in order to progress in some projects. And so far, not in neurosciences, but in health sciences, the public funding reached 220 million euros for a total budget of 460 million euros over the last 6 years. And as you can see here, public laboratories in fact get a huge benefit from these competitiveness clusters. And in France, 2 clusters are really devoted to neurosciences. It's an overlap with the research centers, that is Paris here, and Marseille and South area here with Montpellier. So I think this is one idea we can also share. I will not comment too much on that. That is the Center for Clinical Investigation. Just to mention that the neurosciences is the first round for the CIC activity, covering about 20% of the scientific activity. And what is also important is that we have several centers, but we also have a way to make the centers to collaborate, and this is the Neuroscience Parkinson Network in France, covering different centers in France that are able to set up common research, to set up clinical trial, and to develop a community that is able to work together. One specific aspect in France, that is, we have an organization covering rare diseases. We have the chance to have rare diseases reference centers, and for example, there is one in movement disorder in Toulouse. And we set up Orphan Dev, that is a network for clinical trial for orphan drug and rare diseases. And this is supporting by the Rare Disease Reference Centers, the Foundation Rare Diseases, and FTRAN, that is the European network. And this is important both for academic sponsors, and I can give you here 3 examples from our group, and also for industry sponsor, and also for training. And last few days was in Marseille, a seminar for European orphan drug and rare disease training and education. And you can see here a paper in Lancet Neurology we published on the CMTA research with this type of network. I will not comment on ICM. I have this picture. I think it's interesting because it can show us the merge between here the preclinical research, the industry research, the clinical research, and neuroimaging. That is the main tool we should use to develop this type of collaboration. And in Marseille, we have another institute that is also quite large with the imaging facility, the same type of organization with the Center of Clinical Investigation, like in Paris, that is focusing on different aspects of neurology and psychiatry, and then being linked to different groups that are within this Institute of Neurosciences. And as you can see here, also leading to quite high-level publication and papers. It's good to have local and national organization, but also we have to work together, especially here at the European level. And for clinical research, there is this F-Train, that is a common project between the different entities you can see here, including INSERM, with a shared objective, that is to have a platform and a national access point for both sponsors and investigators. It's not fully dedicated to neurosciences, but of course it can be used to support ambitious academic clinical trials in neurosciences, to enhance the participation and favor coordination of European projects, and also to attract industry to run early clinical development for innovative approaches. And this European group gets quite interesting funding to develop and just focus on 5 axes. So I'm part of the International Scientific Advisory Board for F-Train, and we will support a different network, and there is currently a call that we will judge next month, and there are several networks clearly devoted to neurosciences that are involved in this call. So Europe, and as Etienne already mentioned, I had the chance to promote this big consortium with more than 23 partners all over Europe, from basic sciences to clinical settings and clinical pharmacology. And once more, the objective was and is still, because the project is ongoing, to develop preclinical and clinical model to be able to predict the value of the new therapeutics. And what we intend to do is to develop translatable pharmacodynamic biomarker. And my initial idea was to say, well, when developing a drug, we first start from the animal, and then we try to see and to verify into patients that the results are translatable to patients. And my idea is to say, well, we should integrate all the results, whatever it is on the different method, whatever it is on animal, different models. We are working, I don't have pictures, but on mouse lemur and octodon, as well as in rodents, over rodents, normal volunteers, and early and very specific cohort of patients, and to integrate all this model into a bioinformatic analysis. And I know that India is very well trained on these aspects. And we are bridging all these models with imaging, with electrophysiology, with cognitive testing, and also with development of new biomarkers. And just to focus on the clinical development, we focused on 3 selected challenges, that is trying to induce some deficits in the normal volunteers or in patients that we can correct with new drugs. And so we are developing models of RTMS in order to induce cognitive disorder, sleep deprivation, and we will start in the next few months some hypoxia trial that is also making the bridge between the neuron and the vessels and hypoxia. So I think this was, yes, my last slide. And just to mention, so the city I'm from is on the seaside, and if we are here, is not just to profit, to take profit on the situation, and this is my boat, and so if you come to visit us, I can take you on my boat, but also because we think that there is something important to do to help the patient and science. Thank you.
Centers that are devoted to very rare diseases in India?
No, as of now, we really don't have any centers for the rare disease and orphan drugs yet. We are just in the phase of developing this. Right now, we are actually setting up international collaborative study for looking at ataxia telangiectasia and also looking at other diseases where we are trying to evolve new approaches. We are actually standardizing the ICRS, which is the International Classification for Ataxia Rating Scale, which is not standardized for children. So we just started a program along with Israel and a few other countries, trying to look at these. We'll definitely be interested in looking at other diseases. We have a lot of Charcot-Marie-Tooth and other diseases which are rare, where we definitely need to work at.
Our next speaker is Rupam. Professor Rupam is from the Nizam's Institute of Medical Sciences, and he will talk further on the theme that we've been talking about, is diseases, and in specific focus is Parkinson's disease.
Actually come from an institute which is in Hyderabad, and it's one of the largest centers as far as neurosciences is concerned in the number of students that we produce. We produce about 8 DM students per year and have almost on par with AIMS is about 6 students, I think. But we have about 10 faculty members. Now, we all work on different areas. What has happened dramatically in the last decade or so is that there have been a lot of clinical trials which have come in. Now, this has attracted a lot of both good attention and bad attention. The bad attention, unfortunately, has attracted more of the news than the actual work which gets done. The actual work does not capture the headlines. Just to give you an idea, I'll just walk you through what we have been seeing in the institutions. And we've been all carrying out trials which are strictly regulated according to both your IRBs, institutional review boards, as well as through the central regulatory authorities like the DCGI and the ICMR. So we are quite strictly regulated, and yet in spite of all that, we end up with a notion that all of this is bad. Now, this was somewhere around 2009, when we had a huge number of trials going on in Parkinson's disease, and somewhere along that, quite a few, at one given point of time, somewhere around 12 trials were going on in various stages of completion or otherwise. Some of these protocols have actually started. The first trial was in 2009, which was actually on rotigotine patch. That was one of the first international drug trials carried out in India. And the rotigotine patch worked out very well. It's on the market. And now we have been looking at the subsequent follow-ups on the lower doses of rotigotine patch, the modifications of that. Trials have been carried out in botulinum toxin by changing these are all international drug trials, mainly carried out for both the European as well as the American regulatory authorities as well as for the Indian regulatory authorities. That would mean it's for the US FDA, it's for the European Union regulatory authorities, and hence all these guidelines which require that patients be carried out in both countries in the US and Europe as well as in India simultaneously have been followed. Unfortunately, none of this gets to the newspapers. So what I'd like to say is, what has been the advantage for trials coming to India has been that a lot of patients, can you get this volume up or am I audible at that end? What has happened is that a lot of patients, we as clinicians are extremely busy and we see a lot of patients and patients are much more, have much more faith. I mean, that's the way of conveying, is that they have more faith in doctors here. So when we ask them to consider the fact that they can actually participate in a trial, quite a few of them do so. Now, what it did over the years is that we've developed a close collaboration, and the numbers of patients have been huge. And these have been some of the molecules which have been, I've been working on. Safenamide has been one of the larger molecules which have come out in Parkinson's disease. Is still not released. We're still doing the phase 3 and they've gone for filing. Phase 3 has been carried out in both early Parkinson's disease as well as late Parkinson's disease. This has been done in both the short trials, which are 6-month versions, and the longer trials of 18 months. And initially, the trials, this was one of the largest trials carried out in India on Parkinson's disease. In early Parkinson's, some 300 patients were recruited. In the later Parkinson's trial, something like 600 patients were recruited. It was a huge effort, and somewhere around 80% of the initial trials patients were Indians, especially in the late Parkinson's trials. So these have been presented by many of us, including myself. The primary results have been presented in various conferences and have actually been published. They are now coming out as publications, and these are As you can see, that was the initial first 6 months period, and this was the first 2-year control trial in safinamide. This was initially presented both in Canada, in the Movement Disorders Congress, as well as in the European Federation of Neurology in Budapest. This was in 2011. Subsequently, they were published also. As you can see, this was in early Parkinson's disease. This was published in the Archives of Neurology, and Stocky was the primary author. We were one of the primary authors. Now, what this actually entitled us to do was to see what was these new molecules doing to the patients. If you recruit large number of patients, you can actually see the therapeutic effect much better. You can actually see things happening which may not be visible in 2 or 3 patients. So one of the things we saw was that these patients did not, develop dyskinesia at the end of 2 years, which for Parkinson's is a big thing. This drug was an add-on. It was not the sole drug. And when you do not see Parkinson's patients developing dyskinesia, you wonder whether it's doing something different from other molecules. The second thing which we saw was when he started in late Parkinson's disease patients, you saw actually the patients who had dyskinesias got better, the dyskinesias. Not only did the general Parkinson's improve as far as on-off goes, as far as the UPDR scores goes, their dyskinesias came down. And when you make such observations, you can share it with your co-investigators and sponsors, and they actually designed the second half of the trial to look primarily on dyskinesias. So unfortunately, what happened was that a lot of the advanced Parkinson's patients had very mild dyskinesias. So that was not successful as a primary endpoint. The change was not significant, but when you actually looked at the patients who had severe dyskinesias, they came down significantly statistically. So that has been one of the key points of working in the forefront of a molecule which is being looked at internationally, collaborating with others, and sharing your observations as a clinician. Now, this is something that you don't get to do. These are, as I've told you, is all being published, or in the process of being published. The last one, the long-term, this is actually the original article, and now this is in early, and now the latest ones are in advanced Parkinson's, which have just been accepted for publication in movement disorders. So what has happened over the last 10 years is that instead of being a bystander, instead of seeing the molecules coming down here, you can actually be an active participant, and that has changed the concept of what you do as clinician. In addition to doing standard clinical work, in addition to doing large volumes of deep brain stimulation, you can actually do research which can make a difference worldwide. Now, this trial, I put it up because this was one of the many studies we did, and it was very interesting. What was very interesting about this was a trial which was carried out simultaneously in the US as well as in India. At the end of it all was that there were 115 patients from the US and 64, roughly about 2/3 of that, from India. However, at the end of it, when we actually look at the primary efficacy data, you found that it was working in the Americans, but not working in the Indians. There is a difference in the Indian patients. We are younger. We have most severely dyskinetic patients. But is that a reason? Does it happen every time? Not really. You just do not make the assumption from one molecule. When you actually looked at the safinamide trials, which I told you, which is now actually the top-of-the-line molecule, which is up for filing and should be released in a year or so, when they actually compared the early trials, where 80% were Americans, Indians, with the ones carried out in the US, where 80% were Americans, the data is exactly the same. There is no difference in both the genetic pools or racial pools. And so, to extrapolate from one study that there is a difference is not true. What this led us from clinical trials to clinical research, and Professor Blanc and myself have been working, of course, primarily driven by him, but we've been looking at how to collaborate, how to look at partners from here. Here it was NIMS, along with 2 research centers in Europe, which you can, sorry, this is not very visible, which have been from the CNRS, and you have the Marseille unit, and CISDAG from Montpellier, and Bochum. We had the Ruhr University of Bochum, and they were looking at the biomarkers. We also had a 4th partner, which unfortunately could not be carry on with the project. Sorry. Where is this? How do we do this? There we are. So what we looked at was looking at the scientific knowledge and information exchange, and for the CNRS was the main. We also looked at the biological biomarkers. We wanted to actually develop this, but we had some problems in transferring the samples, which is a major block with both our DCGI as well as the Directorate of Foreign Trade and ICMR. They are very strict about this transfer of samples, even if you want to carry out genuine research or standardize both the centers. That has been one of the major bottlenecks for carrying out anything related to serological samples. And we haven't made much progress there. However, what we've been able to do is that we've set up a lab, and we've been able to actually validate the biomarkers which are commercially available and study tau and phospho-tau in patients with Alzheimer's as well as in Parkinson's plus disease. So we did manage to get something out of it, and we saw that very surprisingly, for a tauopathy, Parkinson's plus, that most of them are PSP, were negative in the CSF for tau, while the Alzheimer's patients were positive. So though they're classified as a tauopathy, we found them to be negative. And of course, we also set up a network with other centers. Sorry, there's a formatting error. This has moved up. Anyway, that was Delhi. It should be down there. All the stars have moved because of formatting. And that was basically with All India Institute. And we had in Hyderabad, we had NIMS. In Bombay, we had Jaslok Hospital. And in Bangalore, we had two centers. We had both NIMHANS as well as we had Manipal Hospital. And we had both neurologists and psychiatrists looking at the neurodegenerative disorders. Basically, we looked at Alzheimer's, and we interact Alzheimer's as well as Parkinson's plus disorders, and we really look to collaborate and develop future effort to look at the biomarkers here. Currently, just to cut a long story short, we put up a couple of projects. Where this has led from the key point of clinical trials to clinical research is now, thanks to Dr. Oliver Blin, we have been able to set up other associations, and we are trying to put up a project for looking at ketamine in the post-traumatic stress disorder after mild traumatic brain injury. We are going to look at that if we get the project. And second one is we are also planning to look at stress cortisol in Parkinson's disease, pre and post DBS, and see whether it actually makes a difference in why DBS has such a profound impact, and whether this cortisol levels as well as BDNF, whether they have an impact in changing the entire course of the disease, which seems to be happening, though there's no hardcore evidence. As a clinician, we can see it happening in front of us. Post-DBS, we see these patients 7 years, 9 years later. They're so much better than they were at the baseline, and they're still staying better. Why is that happening? That needs to be answered. Thank you.
French-Indian collaboration, and we have in the room, in fact, someone from INR, and I hope that you will be convinced by these collaborations. To be a little bit more serious, you showed, in fact, this study with a patient from the US and India with differences. How do you deal with that? And in the future, how do you think that we should do? Should we have less patients but from more countries? Should we try to homogenize the people for the genotype? Should we work on the clinical scale to be sure to have the same clinical scale?
No, the clinical scale was very thoroughly standardized. I have never seen any now, because we've done so many Parkinson's disease trials, the centers which are chosen are actually excellent.
Exactly the same.
Exactly the same. They go through the same rating scale training, and this is double-blinded. They're all centralized, and they're all actually scored. You send in your score sheets on the video you score, and this is actually scored in the US. And the same gentleman who's actually devised the scale makes a lot of money from the pharmaceutical companies assessing your scales, but it's standardized. So there's no problem with the standardization. Now the problem is, when we actually looked at the fipimazole data, we looked at it, there was no standard, there was no protocol deviations whatsoever to explain this. The only sensible reasoning we could give is that somehow Parkinson's disease in India is 10 years earlier. The younger patients are definitely a lot more dyskinetic than the older patients. That has been the uniform observation of all clinicians in India, and they are much more severe as far as the dyskinesia, and their on-off is also much more severe. They do not get so many hallucinations and non-motor symptoms as compared to the older patients who tend to get much more of that.
Are these patients displaying Parkin mutations?
No, not to the best of our knowledge, no.
Questions?
During the last seminar on brain diseases in Barcelona, we connected with Ben Hart, yes, and he is the head of the European Network for Huntington disease, and we discussed with Rupan and other neurologists and the opportunity for Indian neurologists to join this.
Yes.
And Ben Hart, I just sent him an email today, is very keen to develop, not only for Europe, but for the worldwide initiative on Huntington disease, to have Indian centers.
So we should really think to advertise, in fact, for India, because France is often in a strong position, we have to say it, in these networks, and this is something we should do together. So if you are interested, try to, in fact, advertise for including India in this networks. If this can come out from such a roundtable, I think it would be really fantastic.
Actually, what has happened over the last 1 decade is that people in the US and other countries have realized that we have excellent clinicians here. We have no dearth of patients who are willing to collaborate and follow all the necessary regulatory guidelines. What has happened is that we are able to get more patients. So people are realizing they are coming on their own, voluntarily, to bring the clinical trials over to India so that we are able to do this. What we need to show that we are actually able to contribute some more than just carrying out the spine level. Maybe it's the clinical observation. Maybe it's other inputs that we can give in as clinicians that really help to develop this.
Yes.
I'm not a professional neurologist, but I have worked with neurologists and neurosurgeons for nearly 25 years. So I appreciate what you guys have achieved and what you're planning to do in terms of collaboration. But as an outside observer, I think we have an outstanding opportunity to build something, of a great proportions. Another point I would like to raise here is that in India you have tremendous, highly trained workforce of young students who really want to come and do meaningful, clinically relevant research. If nothing else, you have freebies, who can come to your clinics, pull out all the data, and that'll be great help for you. I mean, I think I was visiting Medanta about a month ago by invitation, and that's the same, situation I was presented to me by about 12, 13 of your colleagues. So I think we have all the elements on the ground that can make this thing happen. The second point I would like to make, I was at a conference between ICPO, which is the Institute of Cytopathology and Preventive Oncology in Noida, here in the capital area, and the NCI. And I think India has a tremendous offering for the rest of the world. Size of the patient population, very highly trained, experienced physicians, scientific workforce, and good and willing collaborators in France.
Yeah.
So I think it is just a matter of 2 or 3 months of concerted effort
with Dr. Blind
and blessings, and you will see something happening.
Yes.
Thank you for this very enthusiastic comments that we are all sharing. So we will now switch to Professor François Berger. So he comes from the right side of France. It's in the middle of France, a city called Grenoble, where deep brain stimulation for Parkinson's disease in the Etienne was invented. And he will not be able to invite you to go on his boat, but perhaps skiing, because Grenoble is in the French Alps and it's a beautiful city.
Thank you. Good afternoon. So I will try to give you a short vision with specific example in Grenoble of what could bring technology implanting device for brain disease. There is a strong biomedical need for innovative technology for brain disease to know the physiopathology, but also to treat them. All the targeted personalized therapy progress which were used and observed in oncology were not observed in brain disease, especially neurodegenerative disease, or a very good bad example is Alzheimer's disease. Probably for two main reasons: the low drug penetration in the brain, but also because we do not have access to the fresh brain. We only have access to post-mortem brain, and so we do not have the target. We do not have the real physiopathology. Beside drug pharmacological approach, DBS, deep brain stimulation, so done 25 years ago by Alim Louis Benabid in Grenoble, is a very good paradigm for non-lesional physical intervention in the brain, using what we could name a medtech targeted therapy, so electrostimulation, high frequency, and if you stop electrostimulation, you stop the therapeutical effect, as you can see in these children with dystonia. So it is invasive, it is micro-invasive, but you have no lesion in the brain. And very interestingly, you have a physical impact. You modulate brain function. We don't really know the exact mechanism, but with a physical therapy, you can have an impact on the neuronal function. This paves the way toward a more elaborated theranostic next generation device. And for that, the field of micro nanotechnology and electronics is very important. All this technology were developed for computer, for mobile phone, and now the last iPhone, for example, is at 25 nanometer. So all these devices bring us miniaturization at the size of the molecule, of the cells, but also multifunctional integration, association of several functions in the same small device. Surface modification, nanobeads, and so on. But many publications, many hope. But recently, the European Commission had a very bad statement about the poor validation at the bedside and the few industrial success in the field of smart device. Why? Because everybody forgot that this technology, beside publication, need to be robust, reproducible, safe, low cost, and validated. So we need human-compatible technology. And also it was very difficult to make a clinical development in association with micro-nanoelectronic company, biotech, pharma, imaging, and medical device company. So we really need to develop a new preclinical and clinical strategy for that. For this reason, with Alim Louis Benabid and Jean Terme. Jean Terme is the creator of the Minatech campus, a campus devoted to micro-nanoelectronic, not for health. So we decide to move our research group and a small part of the hospital in this campus to implement a biomedical translational technology center. And the reason for that was to be closer to the technology at the beginning of its translation, but also to meet innovative technology which are not in the field of medicine, because innovation at this level will be very important. So, in other words, to move from clean room to the bedside, and for that, two very important developments: to have a multimodal investigation research center, being able to associate clinic, imaging, physiology, behavior, molecular biology, but also, probably the more important, multidisciplinarity, to have a team with a common language associating biologists, technologists, physicians, mathematicians, and so on. So to do that, we build a new research